Hello students, in today's class we are going to learn about the structure and use of a compound microscope. The aim of the experiment is to study the use of a compound microscope. Coming to the theory, a microscope is a magnifying instrument that is used to visualize objects that are too small to be seen by naked eyes. A light microscope makes use of a visible light, and different parts of the microscope they work together in order to form a final magnified image. Coming to the parts of a light microscope, first of all, we have a light source. Older microscopes used to have a mirror that used to reflect the sunlight towards the specimen. However, most modern microscope contains an electric bulb that acts as a source of light. You can turn the switch on and off for the bulb, and you can also adjust the intensity of the bulb on the scale of 1 to 10 using the knob that is present towards the base. So 1 being the lowest intensity of the light and 10 being the highest intensity. Next is a iris diaphragm. So the diaphragm is present below the condenser lens, and the diaphragm has different sized holes that control the amount of light that can pass through. So there is also a lever present. By moving the lever, this is the lever. So by moving the lever, you can control the size of the aperture so that you can control the amount of light that can pass through the diaphragm and will be projected on the sample. Next is a condenser. So here you can find the condenser lens. The condenser lens accepts parallel light rays that are produced by the illuminator and condenses them into a strong beam. It causes the light rays from the light source to converge on the microscopic slide that we place on the stage over here. And the clarity of the image increases with the higher magnification of the condenser lens. Abbey's condensers are present in most of the sophisticated microscopes with a magnification of about 1000x. Next is the aperture. Aperture is basically the hole that is present in the microscopic stage, and through here, through this aperture, the light is transmitted as it reaches the stage from the source. The stage is present above the condenser lens, and it is basically a flat platform that holds the slide in place using a simple or a mechanical stage clips and enables them to be examined in a controlled way. The specimen can be moved in an X as well as the Y axis by making use of the knobs that are provided for the movement of the stage. Next is the objective lens. So these are the objective lenses which are attached onto the nosepiece. 4 objective lenses are present in a compound microscope. The shortest lens has the lowest power while the longest lens has the highest power. The high power objective lens also is retractable so that when it hits, in case it hits the slide or the stage, then it will retract itself. This is important for the protection of the lens. So there are basically 4 different types of or magnifications of the objective lenses that are present attached to the revolving nose piece. 5x is basically a scanning objective lens, and it provides the lowest magnification power of all the objects. So using this, you can basically scan your slide to find areas where cells will be present. Then is the 10x magnification. It is low power lens, and lower magnification also helps in locating the samples in the areas of the microscopic slide. Then 40x is a high power lens. 40x objective lens is applicable for examination of wet preparations such as hanging dogs, egg and cyst examinations in the stool sample can be done. Then the 10x or the oil immersion lens. This lens is the one in which we make use of oil, which is a medium in between the slide and the objective lens. So visualization of bacteria requires presence of the immersion oil, and it will give a total magnification of 1000x when we use the 100x objective lens. So this magnification is sufficient enough for visualization of fungi, most parasites and bacteria, but it is not enough for observing even smaller particles such as the virus particles that require very high magnification of about 1 lakh x, which can be obtained by making use of an electron microscope. So revolving nose piece is basically the nose piece that holds all the four objective lenses. You can rotate the nose piece, and in this way you can choose the desired objective lens for observing your sample. Coming to the properties of light, when a beam of light basically passes from the air onto the glass, then it bends towards the normal or towards the center. And when it passes back from the glass into air, then it moves away from the normal. This doesn't have major effect when we are using high power lens. However, what happens is that in this process, some amount of information is basically lost. So, however, instead of having air as a medium, if we have oil as a medium between the glass and the lens, in that case, since the refractive index of oil and the glass is same, so the light rays that have passed through the glass and have entered the oil, they will follow the same light path, that is a straight line light path, and hence as a result the maximum amount of light rays are captured by the objective lens, and hence the clarity is more in case of when we make use of oil emulsion lens. Then next is the body tube. So body tube basically transmits the image from the objective lens to the eyepiece or the ocular lens. The ocular lens is located at the top of the microscope, and it is used to look into the specimen. It also further magnifies the image either 10x or 15x times. Usually a microscopic eyepiece has a 10x magnification, and some advanced microscopes have a higher magnification of about 15x. And also most advanced microscope have allow us to observe the specimens with both eyes. Such microscopes are known as binocular microscopes. Then we also have the coarse and the fine adjustment knobs. So coarse adjustment knobs allows large vertical movement of the stage, while the fine adjustment knobs, they are present inside the coarse adjustment knob, and they are useful for bringing about sharp focus of the specimen. So it moves the stage at a very slow rate. So that's it about microscope and its parts. Thank you very much, and kindly like, share and subscribe if you like the content.